Hi, folks. Okay, so this is the last mini lecture of chapter 12. This is 12-6 through the end. I think it's 12-6 through 12-9. I think the hard part's over, so let's clean this up, clean up the chapter, and get to talking about some facilitation and inhibition, which is a lot what's in this part. All right, well, you already learned about myelination, unmyelinated and unmyelinated axons. So now let me tell you that the myelination degree, either myelinated or not myelinated, and then the degree of myelination. This gives us three classes of axons. And type A fibers are a heavily myelinated axon. And because they're heavy myelinated, they are the fastest of the axons, the type A fibers. Type B fibers uh, they're not heavily myelinated. Uh, well, they're myelinated, but they're actually smaller axons. Now, this is something I didn't even talk about before. Inside the axon, when the ions are diffusing, the larger the axon, the less resistance to diffusion there is. So the very large axons have less resistance to diffusion of sodium. So not only does that allow them to be faster, but the myelination allows them to be faster as well. So type A fibers are the largest fibers and they're myelinated, so they're the fastest fibers. Type B fibers are myelinated, but they're not as large, so they happen to be a little bit slower. And then type C fibers are unmyelinated. Now, you saw a video in the last mini lecture that said that the heavily myelinated large axons can reach 100 meters per second conduction speed. And then you saw the movie said that unmyelinated axons rarely exceed 2 meters per second. Well, I don't disagree with that, but I will tell you that the fastest of our axons, the type A fibers, 140 meters per second. I know the movie says approaches 100 or something like near 100, but we have axons that fire up to 140 meters per second. The type B fibers, they're myelinated, but they're smaller, so they're not as fast. They conduct impulses at about 18 meters per second. And then the type C unmyelinated fibers, even though the previous video said rarely exceeds 2 meters per second, I'm going to tell you they're 1 meter per second or even less sometimes, some of these gray fibers. And oh, by the way, why would I call them gray fibers? Because unmyelinated axons appear gray, whereas myelinated axons appear white. And that actually is what gives us our statements, white matter and gray matter. White matter is myelinated and gray matter is unmyelinated. So that's where we get that from. So those are the three types of axons, type A, B, and C. Okay, let's talk more about synapses now. Well, this is the three classification of synapses based on the location of synapse. We can have an axon come down and synapse with the soma. This is called an axosomatic synapse, as you can imagine. An axon and a soma, axosomatic synapse. We can have an axon come down and synapse with a dendrite. This is probably how you've been envisioning this. Because I told you dendrites receive signals, I told you axons send signals. You probably envisioned this right here, which of course occurs. And this is called an axodendritic synapse. And then the last type of synapse is called an axoaxonic synapse. And this can be pretty darn important right here. And it can be because if you recall what this area is the trigger zone. And this axon coming in can alter the trigger zone. It can facilitate it and make it more likely to decide to fire, or it can inhibit it and make it less likely to fire. So these axoaxonic synapses can become pretty darn important in themselves. So those are three types of classifications of synapses based on where the synapse takes place. Now let me talk about this chemical conduction, this chemical synapse. Here's what happens. My nerve impulse is coming down my presynaptic neuron. It's called presynaptic neuron because it's before the synapse. When this depolarization wave hits these voltage-gated calcium channels right here, the calcium channels open and calcium rushes in. Now that becomes important because when calcium rushes in right here, it causes these synaptic vesicles to fuse with the membrane and exocytosize, release their contents, go through exocytosis. So the calcium causes these vesicles to fuse and exocytosize their neurotransmitters into the cleft. Now, when these neurotransmitters go into the cleft, they can be excitatory neurotransmitters, and they cause an EPSP in the postsynaptic membrane. They could be in an inhibitory neurotransmitter and cause an IPSP in the postsynaptic membrane. They can do either or. Usually, we talk about the excitatory ones because if they're inhibitory, then the story is kind of stopping right there. Because now your postsynaptic neuron is less likely to fire, probably won't fire. So that's what's going on here. It could be inhibitory or stimulatory. Well, this neurotransmitter binds its receptors and opens this channel right here. Now, let's say this channel is a sodium channel. If that's the case, sodium rushes in, and that's an EPSP because that's going to depolarize my postsynaptic membrane. Now, if it happens to be a chloride channel that it's opening, that would hyperpolarize that one. That would be an IPSP. But regardless, the neurotransmitter binds its receptor and opens the channel. And if it's at all excitatory, it creates a graded potential in the postsynaptic membrane. Now, before we go into talking about how these graded potentials are all added up, summed, let's just talk about what has to happen in the synaptic cleft and the presynaptic neuron before we talk about the postsynaptic neuron. Well, we got to remove this neurotransmitter. And there's a couple ways to remove this neurotransmitter. One is to break it down by an enzyme, like acetylcholinesterase breaks down acetylcholine. Things that end in ACE are enzymes, and an enzyme that breaks down acetylcholine in the synapse is acetylcholinesterase. If the neurotransmitter was epinephrine, an enzyme called monoamine oxidase would break it down. Oxidase, and et cetera, et cetera. But there is another way to remove the neurotransmitter, and that's reuptake right here. The neurotransmitter is taken back into the presynaptic neuron, called reuptake. And you've probably heard of reuptake inhibitors. Matter of fact, you've probably heard of selective serotonin reuptake inhibitors, otherwise known as, say, Prozac. Prozac is an SSRI. These are mood elevators because they actually inhibit the reuptake of serotonin. And so if you have a selective serotonin reuptake inhibitor, serotonin stays in your synaptic cleft, and that elevates your mood, and this affects your brain and elevates your mood. So reuptake is another way of removing the neurotransmitter. Now, actually, with the case of acetylcholine, both occur. With acetylcholinesterase, acetylcholine is acted on by acetylcholinesterase, and it becomes two molecules. It becomes choline, which goes through reuptake, and it becomes acetate, which is mopped up by satellite cells or just diffuses out of the synapse and is absorbed someplace. So with acetylcholinesterase and acetylcholine, you see enzymatic degradation, and then reuptake, you see both events occurring. And that's what's going on here. Now, there's a lot of really interesting stories in this this chemical synapse. For example, the botulism toxin inhibits the fusion of these synaptic vesicles with the membrane and therefore inhibits exocytosis of acetylcholine. If you can't exocytosize acetylcholine in the neuromuscular junction, then you can't contract the muscle. And that's how the botulism toxin paralyzes a muscle. It actually acts here. All the calcium in the world can come in here. But the botulism toxin keeps these synaptic vesicles from fusing and releasing. So that's something that's going on right there. And by the way, there's lots of pharmaceutical stories at a synapse, just tons, and I could go on all day about them, but just give you a couple of examples, then move on. So that's a chemical synapse, and this is just another way of showing the chemical synapse. It's not as pretty as the previous picture, but here comes my impulse right there. My impulse opens voltage-gated calcium channels, and here comes my calcium inside. Now my calcium causes these synaptic vesicles to fuse and release. Now I have acetylcholine in my synaptic cleft right here, and they bind its receptor and open sodium channels. Now my postsynaptic membrane depolarizes. Now my acetylcholine esterase breaks down acetylcholine to acetate and choline, and the choline goes through reuptake. And that is the cholinergic synapse. By the way, that's a cholinergic synapse. It is a chemical synapse, but it's saying it's more specifically a cholinergic synapse, or deals with acetylcholine. Now, with any chemical synapse, a couple things occur. First of all, you have a synaptic delay. When this action potential gets to your synaptic terminal, your axon terminal, this postsynaptic membrane doesn't depolarize immediately. Well, first of all, calcium has to rush in. Calcium has to cause these vesicles to fuse and exocytosize. Acetylcholine has to diffuse across the synaptic cleft. And yeah, that's only 7 nanometers, but it's still time. The acetylcholine binds its receptors and opens the sodium channels. The sodium then rushes in and depolarizes the postsynaptic membrane. That all takes a little bit of time. So you do have a synaptic delay. Now we're only talking 0.2 milliseconds. That's all we're talking. But it's still a delay, maybe 0.2 to 0.5 milliseconds. Still a delay. All right. Something else that chemical synapses also suffer from are synaptic fatigue. What can happen is these can fire so quickly that the synthesis of the neurotransmitter is the limiting factor. Matter of fact, look down here. Just even speaking of acetylcholine, if you look at the picture 4, choline has to go through reuptake, and then it has to be complexed with acetate again to make acetylcholine. Well, this takes time. And where did you get the acetate? You got it from acetyl-CoA. That's made in the mitochondria, and that takes time. If this neuron fires too rapidly, eventually the synthesis machinery for this neurotransmitter is going to be the limiting factor, and you're going to have synaptic fatigue. You got to quit firing that neuron until you make enough neurotransmitter that it can release and carry out the chemical synapse. So 2 things with chemical synapses: synaptic delay and synaptic fatigue. All right.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open, and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
Okay, that's a chemical synapse. Now, this is another way of looking at the chemical synapse that has just a couple more details than the previous one. That's all.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
All right, so that's the general chemical synapse. Now, these are a list. These are nowhere near a comprehensive list of neurotransmitters. That's not even close. But these are some very major players in our body that we'll talk about this semester quite a bit. You might as well start memorizing them. And when I talk about direct effects and indirect effects, actually very soon here, you're gonna have to start memorizing which ones have direct effects, meaning they open ion channels directly, or which ones have indirect effects, which mean they indirectly open ion channels. But for right now, just look at the list of major players of neurotransmitters. And in addition, I want to tell you that at the last slide of this section talks about inhibition and facilitation. There's more than just neurotransmitters going on here. We also have molecules called neuromodulators. And neuromodulators affect the sensitivity of these neurotransmitters, of the receptors to these neurotransmitters. A receptor could very readily bind a neurotransmitter and very readily open a channel, or the receptor could have a low affinity for the neurotransmitter and not bind strongly at all, and therefore open weakly or not open at all. So we do have these other molecules in our body called neuromodulators. And these neuromodulators fall into two classes: neuropeptides and opioids. And the four opioids, and you've probably heard of probably two or three of these four. You've most likely heard of endorphins. You've probably heard of endomorphins. You might have heard of enkephalins, and you probably haven't heard of dynorphins, but these are all very similar molecules. And these, of course, are pain attenuators, because what they do is they modulate pain signals up the ascending tracts, the sensory tracts going up to your brain. They modulate them and they diminish pain. So these opioids are endogenous molecules that we make that are pain modulators. All right, so those are some neurotransmitters and neuromodulators. Okay, let's talk about this. With neurotransmitters and or neuromodulators, uh, they can have either direct effects, which, by the way, the direct effects are known as ionotropic effects, because they open ion channels directly. Open ion channels. And of course, that's a direct effect. When you open an ion channel, you're either depolarizing or hyperpolarizing, but you're having a direct effect. And some examples, some neurotransmitters that are ionotropic or have direct effects are acetylcholine, glutamate, and aspartate. These have direct effects. They bind their receptor, which directly opens the ion channel and the ions rush in. So acetylcholine, glutamate, and aspartate have direct effects. They're called ionotropic. However, there's a couple types of indirect effects. One's a G protein mediated indirect effect, and one is an enzyme mediated indirect effect. And here's how both indirect effects work similarly. Just one involves a G protein and one doesn't. And here's how it works. Here's your neurotransmitter, and it binds its receptor. So here you go, neurotransmitter receptor complex. It does not open an ion channel directly, not right now. But what it does do is it activates a G protein. And that active G protein, in turn, activates an enzyme called adenylate cyclase. By the way, it could be another enzyme called guanylate cyclase. If it were guanylate cyclase, a similar reaction would occur, but you would take GTP and make cyclic GMP. If it's adenylate cyclase, you take ATP and you make cyclic AMP. Okay? Well, what does cyclic AMP, or for that matter, cyclic GMP, do for our cell? That goes over and opens the ion channel. So you can see that the neurotransmitter itself didn't lead to the opening of the ion channel, but what we call a second messenger. These two right here are called second messengers. A second messenger went ahead and went over and opened an ion channel for you. And some examples of neurotransmitters that have these indirect G protein effects are epinephrine, norepinephrine, dopamine, gamma aminobutyric acid. And I left out histamine just because I was circling the ones that I showed you in the list before. Let me go back and show you something. In this list, look at norepinephrine, and by the way, very similarly related to norepinephrine is epinephrine. epinephrine. There's dopamine, there's GABA, gamma aminobutyric acid. See, these neurotransmitters have indirect G protein effects. And you can see those indirect G protein effects right here with epinephrine, norepinephrine, dopamine, and GABA, gamma aminobutyric acid. By the way, very important word, a word I'll use on the test. Indirect effects are sometimes called metabotropic effects versus the previous effect I told you when you directly open it was ionotropic. So when you directly open a channel, it's called ionotropic. And that's a direct effect because the neurotransmitter receptor complex opened the channel directly. But when you indirectly open the channel where the neurotransmitter receptor complex leads to a second messenger, that's called an indirect effect. It's also known as metabotropic. It's a metabotropic effect. Here's another indirect effect, but it doesn't use a G protein. It's still metabotropic. Anytime you have an indirect effect, it's called metabotropic. But instead of using a G protein, the neurotransmitter activates enzymes. In these activation of enzymes, okay, it's not adenylate cyclase and it's not guanylate cyclase because they lead to second messengers, cyclic GMP and cyclic AMP. But it's other enzymes that lead to other second messengers. And then these second messengers go ahead and open your ion channels. An example of a neurotransmitter that acts this way is NO, or nitric oxide. And that was in our list of neurotransmitters that we previously looked at. If you want to go back and look at that list, there's your nitric oxide right there. So I told you that these are pretty important neurotransmitters, and they have different effects. And there you go. We just about covered that whole list, just talking about some direct effects and two types of indirect effects. Okay, these next couple, 2 or 3 tables, they show you the structure of these neurotransmitters. You certainly don't have to memorize structures at all. What I would like you to do is be familiar with the name of the neurotransmitter and whether it's direct or indirect with G protein, or indirect without G protein like nitric oxide. And so far I'm not finding an indirect without a G protein. It probably won't until I hit nitric oxide. Oh, nitric oxide, not here apparently. You can see that most of these actually work through G proteins. And G proteins are a famous story in physiology. I'm just surprised nitric oxide's not here, but it's not. I didn't see it on there. Okay. So this table, be familiar with it. Start memorizing some of these neurotransmitters and how they act. I would start memorizing them as classes. You can see that they're already in classes for you. This is an amino acid derivative. Well, actually, acetylcholine is an amino acid derivative, as is norepinephrine and epinephrine. They come from the amino acid tyrosine. So I guess I shouldn't classify these differently because of where they came from. You can see that all these down through here are called biogenic amines. They came from amino acids. They're called biogenic amines. I do believe that the reason acetylcholine is an amino acid derivative. Oh, you know what? Acetylcholine is not an amino acid derivative. No wonder. I'm misspeaking on that. Acetylcholine comes from the complexing of choline with acetyl-CoA. It certainly is not an amino acid derivative. Absolutely not. So that's why they classify these differently. You can see that sometimes amino acids themselves, these are just the unchanged amino acid, can be a neurotransmitter. For example, glutamate is a neurotransmitter. It's just an amino acid. That's all it is. I mean, you probably heard of monosodium glutamate. I mean, that's a preservative found in food, but glutamate itself can be a neurotransmitter. And you can see that aspartate, another neurotransmitter, that can be aspartate, another amino acid, that can be a neurotransmitter. We have we have one famously inhibitory neurotransmitter, and that is right here. right here. GABA, gamma aminobutyric acid. There's only two ways to be inhibitory. You open chloride channels to be inhibitory. You open chloride channels, or so chloride rushes in and you hyperpolarize, or you open potassium channels. Potassium rushes out and you hyperpolarize. Both lead to hyperpolarization. That's inhibitory. All right, now let's look up here. Mechanism, location, comments. Direct effects, open chloride channels. Yep, that's inhibitory, open chloride channels. Let me see. Glycine produces postsynaptic inhibitions. Okay, I didn't really. Oh, yep, right here. Opens chloride channels. It was just in a different place because it was under the mechanism of action. So you can see that GABA and glycine have inhibitory effects here by opening chloride channels. And actually, you can see GABA actually opens potassium channels as well. So you can see that. So these are inhibitory amino acids. Not all amino acids are stimulatory. These are some neuropeptides. Substance P is a pain substance. This is the neurotransmitter when you do feel pain. This is how you feel pain. This neurotransmitter is released, and the signal sent to the brain, and you sense pain. Neuropeptide Y, sometimes called neuropeptide YY, is a hunger-inducing neurotransmitter. When you say you stimulate appetite, it's called orexigenic. Orexigenic. Because, well, you know what anorexia is. The prefix a or an means without. Anorexigenic means without appetite. Anorexia, anorexigenic. Orexigenic means with appetite, where neuropeptide YY stimulates your appetite, so you increase your food intake. And then, of course, the opioids right here, they do the opposite of substance P. These opioids, they mitigate pain, they attenuate it, so you feel less pain. All right, so those are some neuropeptides that moderate. You can see that a couple molecules that you've never thought of before as being neurotransmitters are. ATP and GTP themselves are neurotransmitters. I mean, ATP is a energy molecule. It's the universal currency of energy. Well, ATP also acts as a neurotransmitter, as does GTP. And you can see that adenosine, the A part of ATP. Matter of fact, here's adenosine right here. That's adenosine right there. And ATP is this adenosine. Whoops. Well, ATP is adenosine triphosphate. Adenosine is actually just the sugar in the adenine. So just the sugar in the adenine is adenosine. And then if you put 3 phosphates on it, it's adenosine triphosphate. So adenosine itself is a neurotransmitter. And then you see that hormones are listed here. Why the heck would we list hormones on a table of neurotransmitters? And that's because oftentimes a chemical that acts as a neurotransmitter can also be a hormone and vice versa. And I mean that vice versa. Some hormones act as neurotransmitters and some neurotransmitters act as hormones. In fact, I'm going back to show you something. Epinephrine, you know, is adrenaline. Epinephrine is a neurotransmitter, but it's also a hormone called adrenaline. I mean, it's the exact same molecule, really. It just has another name. So you can see that dopamine is also called prolactin inhibiting factor. It's a hormone called prolactin inhibiting factor. So some hormones are neurotransmitters and some neurotransmitters are hormones. In fact, you may take a course sometime in graduate school called neuroendocrinology. And why would we ever teach a course in neuroendocrinology? And the answer to that is because the chemicals are the same. A lot of the chemicals in the nervous system are the same chemicals in the endocrine system. So they have a course, and it's a heavy chemical physiology course. It's heavy in chemistry, but it's called neuroendocrinology because you're studying the same darn chemicals in both systems. That's why. Here's the nitric oxide I was looking for before. I didn't go far enough. This is an indirect effect without a G protein. Nitric oxide and carbon monoxide, neither of them use G proteins at all. So there you go with that. All right, so that's some tables. It's not a all-encompassing, inclusive table of neurotransmitters. It certainly will get you started. Certainly, if you were familiar with those tables, you're a long way to understanding all how these neurotransmitters act. Now, just so you know, you're not doing this in vain. Chapter 18 is the endocrinology chapter. And we're going to do this stuff again. We're going to do G proteins again. We're going to do second messengers again. Some of these chemicals are going to be the same again. So you might as well learn it now because you're going to eventually have to know it later. So, a lot of work up front, a little bit easier later when we do the endocrinology chapter. All right, how do we process all this information? Well, there's a couple concepts here. And the big concept we're going to talk about is summation. I kept saying to you, if you open sodium channels, you depolarize and have an EPSP. If you open chloride channels or potassium channels, you hyperpolarize and have an IPSP. EPSPs are excitatory graded potentials. IPSPs are inhibitory graded potentials. They stand for excitatory postsynaptic potential and inhibitory postsynaptic potential. Well, I told you before that graded potentials don't go very far. They're local, they're decremental. So why the heck have an EPSP if you're not going to go very far? Or why the heck have an IPSP? Well, the answer is this. You don't only have one graded EPSP or one graded IPSP. You have more than one coming in, and the cell sums them. It adds them up. And there's a couple of different types of summation going on. First of all, you have temporal summation, where it sums them in time. Temporal means time. And you have a spatial summation, where you sum two of them that are side by side, not too far. They come in at the same time, but they're side by side. So let's look at temporal summation first. Here's the axon coming in, and it releases this neurotransmitter, and here's a graded potential. Now, before this graded potential completely decrements to nothing, to a resting membrane potential, before it loses any of its depolarization, another impulse comes in. Here's the second impulse coming in. Now, what happened is that depolarizes this patch of membrane, and it wouldn't have hit threshold except that the first decremental graded potential hadn't returned to resting membrane potential yet. It was still partially depolarized. So when the second stimulus came in and hit that same patch of membrane, it added it to the first depolarized patch. That hit threshold, and bang, the signal goes. The action potential is propagated down the axon. It's a temporal summation because we're summing the first stimulus and the rapid successive second stimulus together. It's temporal stimulus. We sum it in time. The trigger zone is always in this initial segment. So that's temporal summation in our decision zone or our trigger zone. Now there's another type of summation, and it's called spatial summation. The two stimuli coming in at the same time, but they're separated by a few micrometers here. Now, neither of these by themselves would have led to an action potential. Both would have been a sub-threshold graded potential. You wouldn't have had an all or none action potential. However, summed together, the depolarization events, the graded potential events of the two together did hit threshold, and this fired, and you see the action potential is propagated. Now, what if you have some IPSPs coming in with some EPSPs? Same thing, you sum them. The IPSPs are going to make you less likely to fire, and the EPSPs are going to make you more likely to fire. Whoever wins, and it's the trigger zone that determines who wins. And this shows you an EPSP and an IPSP canceling each other out. That's what that's showing you right here. So this is a zero-sum game here. But realize that it's not just one EPSP and one IPSP that are being added together. I mean, in this example it is. But you could have 5 EPSPs and 1 IPSP. The EPSPs win. You could have 3 IPSPs and 1 EPSP. The IPSPs win. The bottom line is summation occurs, and a decision is made to fire or not fire based on that summation. All right, so more information processing going on. In addition to all of that, we can also affect the likelihood. Well, not the likelihood. We can affect how much neurotransmitter from the presynaptic neuron to the postsynaptic effector cell, how much neurotransmitter is released to the postsynaptic membrane. We can affect that. And what happens is this. For the main story here is this. The yellow neuron is talking to the purple neuron. The signal's supposed to be going like that. Yellow to purple. But we have a green neuron here. By the way, I'm just saying the colors that are painted in. The green neuron is affecting the yellow neuron. And that green neuron can facilitate the yellow neuron, make it release more neurotransmitter, or it can inhibit the yellow neuron and make it release less neurotransmitter. In this case right here, we're talking about inhibition. So let's talk about inhibition, then we'll talk about facilitation. So the yellow neuron, the action potential comes down, voltage-gated calcium channels open, calcium comes in, calcium makes the synaptic vesicles fuse and release, and I get neurotransmitters released, which bind the receptor and cause sodium channels to open, and you get a graded potential over here. Now, the more neurotransmitter that is released, the more sodium that's going to enter this cell, postsynaptic cell, and the more likely it's going to hit an action potential and fire. Well, how do we have more neurotransmitter released? Well, actually, I'm trying to do inhibition. Sorry, I'm trying to do inhibition. The fewer neurotransmitters that are released, the less sodium flows into this cell, and the less likely that this postsynaptic cell will form an action potential and fire. How do I have less neurotransmitter released? Simple. Here's the axon of my green neuron. Okay, the action potential comes down, voltage-gated calcium channels open, calcium rushes in, calcium makes the synaptic vesicles fuse and release, but they are releasing GABA, an inhibitory neurotransmitter. GABA binds the calcium channels in my yellow neuron. And now when this action potential comes down through, these guys stay closed. They don't allow calcium to come in. If calcium can't come in, then the synaptic vesicles don't fuse and release. If the synaptic vesicles don't fuse and release, then I don't have much neurotransmitter in my synaptic cleft. If I don't have much neurotransmitter in my synaptic cleft, then I don't have much depolarization, and this postsynaptic membrane is not going to fire. This is called presynaptic inhibition. You're going to say, what do you mean presynaptic inhibition? This is a synapse. You are correct. That is a synapse. But the main synapse we're looking at, remember, is the communication between this yellow neuron and the purple neuron. That's what we're looking at. So we're really talking about where is this inhibition, where is this inhibition occurring? And the answer is it's occurring right here, which is occurring before the yellow to purple synapse. So it's called presynaptic inhibition. So GABA is a good example of information processing with presynaptic inhibition. Now serotonin does facilitation. It's a similar story, except for serotonin is not going to inhibit the release of neurotransmitter from my yellow neuron. It's going to enhance it. It's going to facilitate it. So here's what happens. My green neuron fires. Action potential comes down. Voltage-gated calcium channels open and calcium rushes in. Calcium makes the synaptic vesicles fuse and release serotonin. Serotonin now opens more calcium channels here. Well, it doesn't actually open more calcium channels, but it makes those calcium channels more sensitive to the change in voltage. So now they'll open quicker with any small change in voltage. That's what they're going to do now. Well, here comes my action potential. Voltage is changing, calcium channels open, and they're really sensitive now to voltage change. Calcium rushes in, calcium rushes in, calcium rushes in, calcium rushes in. All of this calcium causes these synaptic vesicles to fuse and release. You get tons of neurotransmitters. They bind their receptors and cause this postsynaptic membrane to fire. And this is called presynaptic facilitation. Whew. Now that was a mouthful. So that's presynaptic facilitation as opposed to presynaptic inhibition. And that's information processing.